Should I start?
Sir, yes, sir. You can start now.
All right. Very good afternoon, everybody. All the students who are joining us today for the MOOC courses on Proteomics and Proteogenomics. Welcome to this live session, which is the second live session we have. What we thought, we are going to give you a very brief theoretical background, which will be like a refresher on the mass spectrometry. But then we would like to connect you with our various instruments in the lab and show you live some of the experiments which can be performed using different type of mass spectrometry based platforms. So just to refresh you, which you have already seen in some of my lecture videos, proteomics has been evolving very rapidly. We made a lot of journey, starting from very conventional, traditional protein chemistry, moving to the proteomics. We got from the SDS page, 2D gels, dyes, moving to iTrack, then came the label-free quantification. And then now we are talking about more data independent acquisition based approaches. So it is continuously evolving. What people have realized, how much more robust we can make the field of proteomics, and can we try to minimize the steps? Because in the 2D gel, what we realize that there are too many steps involved, from casting the gel, running the separate gels, doing the image processing, comparing the data, software analysis, a lot of stuff is there. So people came up with much quick and short and the less number of steps, so that less artifacts will be introduced and the process can be more robust. So in this slide, the shotgun proteomics workflow, which involves simply you can take your any of the cells, tissue, any kind of biological samples. Of course, you need to homogenize them, you need to sonicate them, you need to make sure that the cells are break open properly. And then you can add the urea buffer, followed by a step which involve the reduction with diethyl or TCP you can use, or alkylate afterwards with the hydroxamide. Then you want to digest the protein with trypsin or other kind of proteases. And then you are, after doing the peptide preparation, you want to clean up the salt and other impurity which is along with the peptides. For that step, we also use the C18 disk, which is shown here, and that after following up and cleaning up the samples, the pure peptides could be yielded out, and those could be run in the mass spec to acquire data for MS1 or MS analysis, MS2. Then you can do the various type of data analysis with the software like Mascot and MaxQuant likewise. So this slide shows the ultimate impact of the mass spectrometry based proteomics. Over the years, there has been a lot of advancement in the various instrumentation in the mass spectrometry field. But it's still like, genomic sequences were completed in 2001 and 2, but still it took almost 15 years time to obtain the first draft of the human proteome map. But this work was done using mass spectrometry based proteomics to look at all the human organs and can we profile all the proteins from it, which led us to get 70% of the overall proteome of the human. Only draft proteome of 70%. Additionally, other approach of using antibody-based approach led in 2015, which is shown on the right side. That also gave the similar results. So proteomics has evolved, but even since then, lot of data being generated from different type of human samples, which were all as a part of consortium project from the human proteome project of HPP from the HUPO. In 2020, the more stringent data analysis skills were applied. And then from that, we could obtain now 91% of the human proteome. So now we have made substantial progress. On one hand, data quality became much improved and stringent. And then from 70%, we reached to almost 91% of the draft human proteome map. So after this, there are very, quick recap of different type of principles which we apply in the mass specs. I'll just kind of refresh you on that, which you have been studying in the course. Global proteomics, where you look at all kind of proteins. Aim is to look at as many proteins you can try to screen from that, which is also known as many times discovery-based proteomics. If you want to compare your healthy with the disease or control with treatment, that is known as quantitative proteomics. In all of those approaches, you are looking at more unbiased approach to look at the entire proteome as well as maximum type Yeah. So in case of the configuration of the instruments, there are different type of mass spectrometers currently available. Most commonly used configurations are have been Q-TOF in the past, but now we are using more of the Orbitrap instruments. Of course, Q-TOF are equally used as well, but Orbitrap gives slightly higher resolution. So what we see here for the data dependent acquisition or DDA is having the quadrupole along with Orbitraps. And then we also have the instruments like quadrupole followed by the time of flight. Additionally, different type of triple quads are available, which is being used for the targeted approaches. So there are different type of mass spectrometers available, which you have already been studying in the course. I do not want to take a lot of time on doing that. And then, of course, if you are looking at more quantification approaches, then you can look at tandem mass tag or isobaric tagging for relative and absolute quantification, or you can also do the label-free quantification, no labels being used. More recent approaches are coming based on the data independent acquisition, which is giving much more higher coverage based on the libraries. So during the COVID time, we did a lot of research, and that's for the time which we realized that how these technologies with so much unbiased approaches can give us a lot of information. And while for many other type of biological question to be addressed, we need antibodies and many other reagents, which takes its own time to be procured or to be generated. But in case of the mass spectrometry based technology, one could do many tests and assays without having a lot of requirements for the reagents. So one thing which we try to look at was uh, when the patient's nasopharyngeal swab samples are taken, which was used for the real-time PCR analysis for the COVID-19 detection. Can we take the same sample and are we able to see the viral peptides? Given that this is all human sample, what we are talking, and viral peptides will be in a very trace amount, very minimal amount. So can we try to still look at all the peptides coming from the virus? So when we try to extract with different type of organic solvents, ethanol, acetone, isopropanol, mix them together to get lot of maximum peptides from virus. And after running the sample, of course, mass spec can acquire all the data. But after the entire data acquisition, when you search the data with different type of databases, you can get different results. For instance, same data we analyze with the uh, SARS-CoV-2 database to look for the viral peptides. And then we analyze the same data with the human to look at all the human proteins, right? But fortunately, we could see many viral peptides coming out of it. Based on that discovery workflow, which we use on the Orbitrap fusion, then we try to come up with the MRM-based assay based on 11 peptides taken from the spike protein, nucleoprotein, and replicase polyproteins from the SARS-CoV-2 infected individuals, COVID-positive patients. So this just showed you like kind of utility of these mass spectrometry technologies even at the time when we need it, and also their unbiased approaches. What I'm showing you right on the screen is the one of the or two of the most recent mass spectrometer instruments, which are currently being talked in the proteomics industry heavily. One is the Astral Orbitrap, recently launched by Thermo Fisher Scientific. Another is the timsTOF Pro 2, which is launched by Bruker. Both of these are the highest end mass spectrometer. I would not say which one is better than other. Of course, you can use it and share your results with us. But these are both the best mass specs currently available to give us the data at very unprecedented scale, which we were not able to procure earlier. This is the Orbitrap fusion, what I'm showing here. To save time, I'll reduce the animation. So here in the fusion, you have the quadrupole, you have, you got Orbitrap and ion trap, and you can generate variety of data sets from that. We are now going to take you shortly to our mass spectrometry facility, which is another national facility for high resolution mass spectrometry based research and training facility, generously funded by Department of Biotechnology and Ministry of Human Resource Development. Also, we have a COE from Thermo Fisher Scientific. You see in the center, we have the Orbitrap fusion, which we'll have demonstration shortly. On the right-hand side, we have the Q-executive Orbitrap mass spectrometer. And on the left side, we have the triple quadrupole ALTIS mass spectrometer. So all the three mass spectrometer, we will have the live demonstrations from the lab. Very briefly, you can do a lot of things on all of these mass spectrometer, but we will try to focus more on the Orbitrap fusion for looking at the deep proteome analysis, how you can try to get maximum number of peptides from the given sample, and can you compare them across from the control versus test condition. So that is more like deep proteome analysis as well as quantitative proteomic analysis we'll try to show from fusion. We'd also like to talk to you about Q executive mass spectrometer, which we are employing with the UHPLC to do other application, not proteomics but metabolomics. And they both go side by side to add a lot more, multi-omics data set. So we will briefly touch upon the metabolomics workflow using the Q executive orbiter mass spectrometer. And finally, we'll show you the triple quadruple ALTIS to do both, the quantitative work on the targeted proteomics, and if time permits, we can look into targeted metabolomics as well. So again, these are the various type of mass spectrometry setup. I don't want to make it the theoretical class again. So we are now going to take you to the our lab directly, and one by one, TAs are going to start from the fusion, Q executive, and then ALTIS. So now I hope you're ready for going to the lab and starting this session.
Okay, so good afternoon all. So now we are standing in front of the orbiter fusion, as you can see over here. So orbiter fusion is a hybrid mass spectrometry, which having a quadruple orbiter and so how this particular system actually has connected. So this is a nano LC. This is a liquid flow system which uses the binary, which having a binary solvent system. So there is a solvent A, which is a 0.1% formic acid, and this is the solvent B, which is a 80% AC and 0.1%. So these particular two solvents actually come these two tubes, one for the solvent A and one for the solvent B. And over here, there is a pump is there that helps to mix the solvent. And this particular solvent, along with the samples, goes from this particular tube to this particular column. So now let's see how the samples are present. So this is the nano LC system with the auto sampler. So this each small vials, as you can see, for example, this particular vial, this each vial includes the samples over here. So as you can see over here, these particular vials. So over here, the samples are there, and there is a needle inside that particular sample. So this particular needle actually injects, takes the sample from this particular vial and injects into the particular column. Now, how this particular actually columns look like? So this is how a particular column, looks like. Okay, this is a particular column, how it looks like. So over there, this from here, as you can see, these particular tubings are getting connected, and from this particular connection, the samples is going, and this is the spray. So if I use this particular thing, if you can focus over here. So this is the particular spray where we can see that the needle is there. To make these things more easier for you to understand, so we are showing over here how a particular needle is very much small and very thin, which cannot be seen using the naked eye. So that is the reason we have a camera over here that helps us to understand that from this particular part there is a spray is happening, and this particular spray is actually entering into the mass spectrometry to this ITZ, which is that helps you to get the spray inside the mass spectrometer, where the ionization start using the different gases over there. So now this is from here after mass from this particular mass spec. When acquisition happens, we start generating the data. Now once we start generating the data, this we will see that how a particular chromatogram looks like. So now, currently we are showing the actual chromatogram how it looks like. So once a particular peptide start eluting and it enters into a mass spectrometer,
as you can see, it's a gradient of 2 hours where a particular sample start eluting from somewhere around 5 minutes, and you can see each this particular dense chromatogram, which includes each of the peptides as it comes from once it elutes from the particular column. So now we will see that how this particular each of these particular precursor look like. So as you can see, this particular peak is actually, for example, this 6.23, then followed by 6.28. This actually represent one precursor over here. Now once this particular precursor is entering, now we can also see how the MSMS is actually look like. So the first one which we are seeing is the how the peaks looks like in the MS. Now if I am just clicking into this particular part, you will see that this is how the MSMS is looking like. So as you can see over here in the bottom panel, there is each of these MS has been then further fragmented into the MSMS. So this particular each MSMS is actually helps us to identify the transitions of the amino acids, and then finally we are taking this thing forward. We can understand that what is the peptide has been identified. But right now, whatever I have showed, and like if I look into this particular chromatogram, we will hardly we might understand the quality of the data, but we hardly understand what proteins or what peptides gets actually identified. So this actually comes in the format of a raw file. Maximum. So format of a particular raw file. So these particular raw files are mostly the structured or the mostly the unstructured data, and then we have to take it forward for doing the analysis so that we can start identification. So right now, as you can see over here, so once we get a particular raw file, this particular raw file comes from the particular peptide, and this particular peptide actually we get the MS spectra. Now, what do we have to do from this MS spectra or from this particular chromatogram? We have to go back to the identification of this particular protein. So for that thing, so there are multiple analyzer and strategies are there which generally people uses. So the first thing is that from the spectra that we have shown, that is the experimental spectra. But to understand what particular this experimental spectra is actually given, we have to compare this experimental spectra with the theoretical spectra. So the theoretical spectra over here we get from the database or the human database that we actually uses for the analysis. So as you can see, this MSMS spectra, the theoretical spectra, and the this experimental spectra actually gets compared, and it gives the identification of each of these peptides. And followed by when multiple peptides gets identified, it's finally come forward and we start understanding that these peptides align with which protein sequence, and then the proteins get identified, as I have shown over here. So now one particular peptide actually from one particular spectrum, multiple peptides can be identified because it's based on the prediction. So the one which is having the maximum score actually taken forward, and from where protein sequence get identified. So there are n number of softwares are there which you can use for the this analysis of this particular mass spec raw files to identify the proteins. But the one which is globally people has used and having the maximum user is the MaxQuant. So MaxQuant having the search engine which is Andromeda, and is one of the user friendly free software available in the internet. So once you download the MaxQuant, as you can from here, this is the MaxQuant official site. Once you download it, you will see a MaxQuant icon over here. So once you click into this particular MaxQuant icon, it gives the interface that somewhat looks like this. So now the next part is that the loading the parameter file. So once you load the parameter files, then you will have the all the parameter which is required for doing the analysis. For example, I will show few of the parameter, but for example, you use trypsin for doing the enzymatic digestion of your protein to convert it to a peptide. So that trypsin needs to be added over here to make sure that the theoretical spectra that gets generated based on the cleavage size that trypsin gives. Okay. So that is the first thing where we are adding the parameter. The next part is the raw data. So the raw data means the file that I just show now, the chromatogram file, which is a unstructured, not readable data, need to be converted into a structured format and a readable format so that we can see and understand that what particular proteins has been identified. So this particular raw data can be, we can add this raw data once we, once we have the particular files. So this is, simple, a general, a screenshot I have taken, which I am just showing that what are the files you will be requiring. So this is the, data from the COVID manuscript which we have published in 2020. That we have, these are the raw files we have to take. We have to take the maxquant parameter file, and we have to take the database. So at first we have to load the maxquant parameter file, followed by loading the .raw file, and then loading the database and click start. That much simple maxquant is. It just requires 4 to 5 steps to start any analysis if you have the raw files with you. And coming to the, some of the simple parameters, that if your raw files are not fractionated, I think as you have gone through the, videos, you are aware that what is fractionation and fractionated samples are. So, but most probably in the different kind of analysis where we generally go for in solution direction, and we don't usually prefer to do the fractionation. So for that we have to make sure that we are clicking into a no fraction over here. And the next part is that like what is the total number of processor you want to give, so that your maxquant runs according to the compatibility and the number of processor that you have in your PC. Now the next part is that once the analysis of the maxquant is done, you will get this particular combined folder. So once you get this particular combined folder, from this particular folder you will get the text folder here, and from this text folder you will get the protein group. So this particular protein group is something, when you open it, you will get the protein identification. So just to take everything starting from the the mass spectrometer instrument, we have shown that how a peptide sample which you prepare, the particular mass nano LC takes that particular sample, spray the sample into the mass spectrometry. Mass spectrometer using the analyzers you are using, maybe quadrupole and Orbitra, that helps you to give the MS and MSMS spectra, that gives the black dense chromatogram that we have shown. And that particular chromatogram, next it will help, it's the unstructured data, and finally you take that particular data forward for analyzing and try to understand that what proteins groups or what proteins you have got. So this is the way you can take a particular samples and finally identify the number of protein. So this is about the proteomics sample analysis using mass spectrometry. And I think now we will move forward into the next instrument, which is Q-exactive Orbitra. And we will show that how Q-exactive Orbitra, which is another hybrid instrument, can help in the identification of the metabolomics, or can be used for the metabolomics experiment. Okay, so now we are in front of the Q-exactive Orbitra, and Ankit will be giving a demo of this particular system.
Yeah. So hello everyone. Good afternoon. So now you have seen that you have done a proteomics experiment using a fusion, orbital fusion, using the tri-bit mass spectrometer. You connect it to a nano LC. But here in the case of metabolomics, what we are doing is that we are connecting it with the HPLC. The major difference between the HPLC and the nano LC is the flow rate. So here we can go to the a very high flow rate. Now here, as you have seen in the nano LC, there are two solvents. So it is also going for the two binary solvents to form the gradient through which the compounds can be eluted out. So now here is that you can see that there are the pumps through which the solvents get mixed with, and here it is the position where the samples are kept, which is the auto sampler. So you can see that the vials, generally this kind of vials are generally put into here, and here through the auto sampler, when you just define the position, so what it does is that the needle comes and injects it automatically and takes the compound. Now the sample will go into the column. So you can see that it is a column with the material on the basis of the compound you are on the compound and the chemistry of the compound that you are trying to separate or elute out. So you will use that particular column. So for example, here if you want to elute out the hydrophobic or test out the hydrophobic compound, so you can use a C18 based column, which will separate on the basis of the hydrophobicity of your molecules, which will elute out on the basis of the gradient that you are uh, doing, and then finally it will go to the into the instrument, through which after ionization it will go into the mass spectrometer. So here in this instrument, exactly there are the two kinds of mass analyzer. One is your quadrupole and then is the orbit gap. So here, if you know that in the protein, which we need to have a mass analyzer or to get the tandem MSMS spectra for identifying of your peptide sequence, but in the case of metabolomics, we can get the M by Z of a particular molecule. But the second MS fragmentation spectra helps you to get out the spectra or the fragment specific spectra for that particular compound based on the coordination energy that you provide. So now I show you the one gradient and then how it is set up and how we can use it.
Yeah, so still Ankit is showing the gradient. This is the display panel of the mass spectrometer. So we can understand from this particular panel that how the scanning is happening. So right now we will try to look into the gradient of the metabolomics data.
Yeah, so as I was mentioning, that we can use there is the solvent A and B, through which it gets mixed. So for that we need to specify the instrument that how much percentage or how much hydrophobicity on the basis of their compounds chemistry you want to use. So here you can see that here the gradient is going from 5% of the solvent beads to getting increased to 98% of the solvent, and then it is so in the basis of the hydrophobicity, your compounds will get eluted out, and then finally you can get analyzed in the instrument. So now another thing is that, so as I was mentioning, that if you need to know the M by Z or the mass fragment, then you can do it in a single MS. But if you want to go to know the spectra and the fragmentation pattern and whether that compound is actually that you are analyzing or not, so you will go to a MSMS fragmentation. So for example, another in the concept of the targeted proteomics is your MRM and the PRM. So here in the Orbitrap, if you want to do a PRM based study for for a particular precursor, for example, if you have done a discovery based metabolomics study, as you do for in the proteomics study, that you have identified certain metabolites that are getting very much dysregulated in your particular cases of the disease or a particular case of your study or interest. So now you want to validate that the compounds that you are looking for that is actually that particular compound. So one strategy for validating those is the PRM based or the parallel reaction based monitoring of those particular compounds, as is the case of the peptides. So for that, what we need to do is that you need to define the precursor. So what you will do is that you will say that the mass molecular weight, according to the molecular weight of the compound, you will define the molecular mass for that particular compound. And then on the if you and in the metabolomics, if you can analyze in both your positive mode as well as your negative mode, that is, the charge can be at the M plus H or M minus H. So if you are analyzing in your positive mode, so what you are doing is that so it will you will give the instrument the precursor molecule. So it will filter out that only that particular compound having that particular M by Z. and then this precursor will be fragmented at the MSMS level. So after you have assigned your precursor value, so then it will get fragmented and all the fragments will be captured at the MSMS level. So now you can see that, for example, this compound reserpine, that M plus H precursor you have defined as 609.2870 at M plus H. So M by Z is that much, then it has got the fragments. So now there is a fragmentation pattern generally that is being that you can identify from either from your global MS2 based studies or from the repositories like your HMDB, MONA, Mass Bank of North America, Human Metabolome Database, etc., etc. You can go and search that there are the fragmentation patterns which have been reported by various labs across the world who have done for the fragmentation pattern for that particular spectra or the fragmentation of fingerprint for that particular molecule, and that is being there. So after you have given in this software Skyline the different precursor fragments, so this becomes then the transition, that is the precursor and the product ion. So then you can see the peaks. For example, if I show you, so here, yeah, so you can see that though the peaks, which is the place where it is getting eluted out, the molecule is eluted out, the retention time of the compound where you can find out, and the fragments which are coming out. Yeah, so here you can see that the molecule, the fragments, yeah, the fragments like the at the 610 and the precursor and the fragments that are getting fragmented at particular the fragment ions at the particular peaks. So this, in this way, the transition is performed, and then you can try to quantify and find out that how that you can validate that particular molecules. So this is how you can perform the global metabolomics and the targeted molecule metabolomics in QExactly. Okay, thank you.
So we'll now be moving to the targeted proteomics part. So for this, we use the triple quadruple mass spectrometer. I request the TS to take over.
Unshare कर आवाज नहीं आ रहा.
Which is 12 portable. So now I will show the parts of the experience. So we have the key compartment here. So in the first we have different buffer system. So here we have quaternary pump. That means we have two solvents, and one is for middle wash, one for solvent. So here we use solvent A and B. The solvent A is you can see pure in this water, water, and solvent B is HCl. So after that, all the solvents come to the tube. So here we have the quaternary pump. So quaternary suggests that four functions which can here and mix here. So this is all the pump that we have. So after that, here is the auto sampler we have. So here we can put many samples. So this is the vial where we submit our we have different types of vials.
Column. এটা column.
So here also we have different using the station will automatically detect the
Column দিতে হবে। ও আচ্ছা, এবার বুঝেছি।
This is the column compartment here. So I can open and show you the column compartment. This is the column we have here. So this is the column that for our here is
মানে এটা কি তোমার রেজাল্ট ফাইন করেছিল?
Good afternoon, everyone. So, this is Medha Gayatri. So, I'll be showing you how we take the targeted analysis ahead. So, as shown by Shashwati, right now we this is how we run the instrument. It has an HPLC, and after running the samples, HPLC connected to the triple quadrupole. We keep the samples in the mass spec, and so the approach for targeted proteomics is a bit different. So let me start by sharing my screen, just a second. So in targeted proteomics, to ensure that the instrument is working fine, we have a few standard samples that run every day in the instrument. So we check how the response is coming every day. So this is a pre-optimized list of peptides that are just monitored every day, and we inject the equal amount of this peptides. So these are like proprietary peptides obtained from commercial companies. It can be anything. Like here in this case, we are using a mixture of pre-labeled peptides from Thermo and BSA. So this we have like this we have multiple aliquots, and we inject each amount each day equal amount. So you can see the response more or less remains same. So if we start from 31st August to 13th September, you are seeing that there is more or less equal response. And for the past 2 to 3 days, we have seen a bit of slight increase in the response. So this is a line graph has been drawn. So to monitor this day wise QC checks, we call it QC checks. We use Panorama Web. So this is a freely available portal based this thing. So we can upload our data on this, and it will automatically do the analysis and let us know how much the peak areas are coming for each of the peptides that we have injected, and accordingly give us a plotted graph like this. So this is one way that we monitor the instrument health and how it is performing. So apart from this, one major concept that everyone trying to learn targeted proteomics needs to understand is that in targeted proteomics, it is different from global proteomics. In global proteomics, we are directly starting with running the samples and then analyzing the raw files and seeing how it is what proteins are coming, right? So but in targeted proteomics, we have whatever we have a list of proteins. Just let me just explain. I'm sharing my screen. Yeah. So just going a step back and trying to understand, like in global proteomics, we are not giving the instrument what needs to be analyzed. That is defined based on the MS, the ability of the mass spectrometer, the resolution, and all other parameters that we are using. But in targeted approaches, we tell the instrument what type of precursors we are looking for. So there are many types of targeted approaches. Mainly, we'll be talking about MRM today. So in MRM, it is more about you give the instrument. MRM is usually done on a triple quadruple mass spectrometer that you just saw. And in MRM, you usually try to see like you give the instrument the precursor and product ion charges, and based on that list is what the instrument gives us the data. So before running the samples, you need to give the instrument a list, or else it won't be able to run the samples. And then based on that list, you analyze your data. So just a very basic understanding of what is SRM and PRM, the difference. So in SRM, usually we use a triple quadruple mass spectrometer, and there is ion selection at the first quadrupole and also on the last mass analyzer. And after that, we get this kind of peak. So, but it is different from PRM, because in PRM we only tell the instrument the precursor ion that needs to be selected. There is no further selection done from our end after this. The instrument does it on its own. So it's an orbit trap is used instead of a quadrupole, and then we acquire the data. So apart from the difference of the orbit trap and the quadrupole, and also the resolution at which both the techniques operate, both the basic principle of both SRM and PRM remains the same. So how the data is acquired, you might wonder. So we have a a table like this. I hope it is visible. So a table like this, where we are giving the instrument the precursor Q1 charge and product ion charge, that is Q3 M/Z, and we are giving at which collision energy it should do the collision of this particular precursor to see this product, and how much time each product ion gets to be analyzed in the mass spectrometer. So this I am showing for an SRM or MRM. Words are used interchangeably most of the time. And once for example, a peptide of this length is breaking, and then there are multiple Y ions and P ions. And out of that, a few of the Y ions we will be monitoring, whichever we find from earlier experiments were coming good, or which were seen to be coming good in our global data. So for a transition, like for example, a pair of a Q1 and Q3, that is a pair of M/Z for first quadrupole and the third that we are giving, that is called a transition. So each transition, if we give 250 milliseconds of dwell time, or time that the instrument will take to acquire data for this transition, the total will be into number of that. So every transition will get that much amount of time, and the time required to acquire one data point will be 1 second, because it will be 215 to 4000 millisecond 1 second. So like that. That is how the kind of like the data acquisition is done. So I'll explain that here again. So at per second, these are the for example, if for this peptide, these 4 fragments we are monitoring, at the first second we get these 4 data points, and similarly, like at each second we'll get more and more data points, and so this peak will be formed. And now you understand how we see these peaks that we have been showing you for SRM or MRM. So basically our data is a collection of data points at different RTs for different fragments, and then they are joined to form a peak shape. So reliable peak identification is done based on various parameters, which take a bit of more time to explain. We will just move on to which particular software we use. So there are many softwares that you can use for targeted data analysis, but we in our lab usually use Skyline, which is a very freely available and open source software. It is very user friendly, and right now, like, I'll just directly go to the software. So if you want to look for the software, you can just type Skyline MS, and it will take you to the their project page, and this is the project page, and you can download the software from here like any other standard software. I'll just like to show you how a particular blank Skyline document looks like. So whenever you have installed your Skyline and you have this, you open a blank Skyline document, you will see that here this panel is where all the targets you want to monitor you will feed them here, and then there are various settings. So here you have to take into account mainly peptide settings and transition settings. So based on what parameters you want to filter out your peptides. So basically trypsin cleavage and the background proteome, we will add the human background proteome. So this you can make, I have already prepared the proteome here, but you can make it based on the human FASTA sequence from UniProt or any other source that you have, or depending on the organism that you're working, the background proteome will change. And then you go to the filter tab, and you see here we filter which peptide length we want. So based on, if you want smaller peptides, so like usually 8 to 16 is a very a good peptide length. And then if you want to exclude any peptides having any kind of modifications, you can exclude them. If you have a library which is prepared from runs of the your experimental data set, or from repository data set, you can add that library here. And you can mention any other modifications that you have introduced into your samples of peptides. So this is important when you are running some synthetic peptides where your N terminal or C terminal has been labeled. There you can add the isotope modifications here or any other structural modifications that have happened. So in this case, we are just showing everything, so we are not going to add anything here. We use the software for both pre-run preparation and post-run analysis. So before running, we need to prepare a transition list that I have been showing a box of Q1 and Q3, and that is prepared using this particular document. So I will just show. Just a second. So for example, these are 4 proteins that I want to monitor. These are the UniProt IDs of 4 proteins that have come up significant in my data. So what I do is I take the UniProt IDs, I copy them, and once I have, if I'm sure that I have the here I have put the background proteome. So then I can just go to edit, insert, proteins, and add the protein IDs here, and it will directly map in the background proteome to the protein name and its sequence. So once I say insert, based on all the settings that I have put here in the peptide settings, it will automatically give me the highest possible peptides that will come good in our data. So that is how we get the list. Now what you have to do simply is you have to export the transition list. Sorry, you have to go export transition list, and based on the instrument and the parameters that you want. So here we are using a triple quadruple thermo altis, and we need a We prefer around 300 300 transitions per list. So it will come up to, because currently we have 1512 transitions in this list. It will come up to around 6 lists. And once that is done, you use, depending on the gradient, you tell it this, and it will export. And you say save. So I already have that saved, and I'll just show you. Okay. So basically we get a list like this. So this is a transition list. It will give the start time, end time, the precursor M by Z, product M by Z, and the collision energy, as I showed. The isolation list is used for PRM, and it will just have the precursor M by Z and the collision energy. That's it. So this we feed into the instrument, and the instrument runs, and we get the data. And we use Skyline for similarly after this to analyze the data. So this was a very brief of how we do targeted proteomics, but I'll just close this here, because that much was the scope for this today's session. Thank you.
All right. Thank you very much, TS, for giving the live session from the lab, showing the instruments. And as the participant will appreciate that different type of principles which we have been talking for various type of mass spectrometer, you have seen all of them at once now, whether it is quadruples, orbitrap, ion trap, triple quadrupole, all of these principles we have been talking, and those were in these mass spectrometers. And we also showed you ionization source like electrospray ionization. In this particular lab room, we don't have the multi, but we have multi in another mass spec lab. So what is more important to emphasize here, that irrespective of where you are working on right now or what kind of application you have, you will be in some or other way benefited by learning mass spectrometer, because you will be using it for some other application. Essentially, if you think about even when you are coming for protein purification based work, you need to identify the protein. Or if you're looking at any kind of interaction map, you need to do the pull down assay, again identify the complex mixture of the proteins. Or if you're looking at comparative changes, differential proteomics to compare disease with the healthy, you need to identify large number of protein followed by their quantification. And eventually the goal is now how to validate the protein without availability of the antibodies. And that's where Mitha was talking to you about MRM and PRM approaches, where you can use these type of, for MRM, you can use the triple quadrupole approach, or for PRM, you can use the orbitrap approach to validate your proteins of interest, because now you have identified the peptides. You can focus on those peptides and you can try to validate those further. I think some participant had asked question that whether mass spectrometers can be used in the clinics, right? And that is something very relevant comment, because lot of emphasis going on one hand for R&D purpose, research development purpose. Many mass spectrometers are currently available and they are being used. They're quite costly equipment, very big setup. But when it comes for the taking to the small miniaturized mass spec to the path labs and clinical setting, there is a gap is still available, and that's why there's a lot of work going on. And the MRM type of assay, which is working on the triple quadrupole based mass spectrometer, they are closest going to the clinical assays right now, when we are looking at many type of clinical tests being developed, which can be utilized for looking at potential biomarkers for early screening of diseases. But there is a lot of emphasis going on how best one could miniaturize these mass spectrometers and then try to apply them in the path labs and clinical settings. Additionally, as what we are talking to you about mass spectrometers, which is giving you only a stretch of peptide sequences, right? We are not getting the whole protein sequence per se. So there's a big emphasis going on. Can we do the protein sequencing the way we do the next generation sequencing of the genome? Similarly, can we have the next generation proteomic sequencing technologies? And you'll be delighted to know that there are many good approaches coming forward from different companies which are based on the good scientific R&D work done. And maybe in 5 years' time, we will have some good protein sequencer available, which can do the protein sequencing as well. So the lot of excitement happening in the field of mass spectrometry and proteomics currently. And I'm sure as you are learning from this course, you will see that now you have better ability of looking at data and the raw data at a much wider scale, at much higher magnification level, because you can now take the same data and you can analyze the data with various type of software tool which we have been talking in the course. You can take the mascot, you can take mstrack pipe or maxquant. You can look at all the protein changes. You can look at a specific quantification analysis from the metabolist or Perseus. You can now look at specific peptide targets using the skyline, which we just showed you right now. Many open access tools available which you can utilize. All you need to do is access of good mass spec raw data from your own data set or from the publicly available repositories and data set. So now you have sky's the limit. You can do a lot of analysis. And when you do analysis, you have better idea for how you have normalized the data, what was the artifacts, what is missing value imputation, and then you can do better justice to the analysis and the outcome coming out of that. I hope you have found the proteomics course useful, and it has been at least triggered as a catalyst for enthusing you towards the proteomics journey. We will be, of course, available to you to train you further in this area if you need further assistance from it. I think after this proteomics MOOC participant will not be there live. So wish you good luck for your exams, which will be upcoming soon. And some of you who will be toppers of the course, we will be happy to take you forward either in our lab for some internship or give you opportunity for attending some workshops which we'll be conducting. So good luck everyone, and we'll be happy to keep interacting with you. On behalf of all the TAs who have really worked hard for doing things in time, let me sign off from here. Thank you everybody. Bye.